So, guys, in the second part of this chapter, we have another 10 questions. So, in this question, it says which process requires energy. So, we have five answers. One of them requires energy, the other four they do not require any energy. We have passive diffusion. You know that diffusion is the movement of molecules from high concentration to low concentration that doesn't require energy. Facilitated diffusion of calcium through a channel. So facilitated diffusion, it's like diffusion, but it's happening through a protein channel. Okay, again it's from high concentration to low concentration that doesn't require energy. Water flowing into a paramecium in a lake. So guys, we have, let's say we have a lake. Okay, this is a lake that contains high amount of water, and we have a paramecium, which is an organism. Okay, so or this paramecium is very small and it contains a little bit of water. Okay, so when water moves from the lake into the paramecium, okay, this paramecium, then it doesn't require energy because water moves from high to low. Okay, which is by osmosis, by osmotic pressure. This will happen. So we don't require energy for flowing water into a paramecium. We have a contractile vacuole. Okay, it's one of the vacuole that contract for removing water from an amoeba. So guys, we have amoeba, okay, that contains a contractile vacuole. In order to remove water, okay, you know that inside the cell there is low amount of water when compared with the outside. Okay, so in order to remove water from low concentration to high concentration, it needs energy. So this process, it requires energy. Let us read the other option that we have. Osmosis of water in a cell. You know, we know that osmosis is a type of passive transport that doesn't require energy. So I hope all they are clear, and the answer for this question is D. Okay, D is our answer for this question. Let us move to the next question. The ability of, or the ability to respond to stimuli. Stimuli, it means any physical and chemical thing. Okay, physical like touching, or when the sun exposure into our eyes. Okay, all they cause, all they are regarded as stimuli, and they cause a response from the body. Okay, when we look to the sun, guys, the sunlight is a stimuli, but it's physical. An example for the chemicals, when we are eating food or anything that it has a smell and it has a taste that we can feel. Okay, so all these physical and chemicals are regarded as stimuli. So what we called it, response to the stimuli, we called it irritability. Okay, irritability is, it means responding to the environment, to the physical and chemical things. That's one of the characteristics of the living organisms. Okay, locomotion does not have any relation with the responding to the stimuli. Locomotion, like movement, characteristic of nerve cell only. It says yes, one of the important characteristics of the nerve cell, they are responding to the environment. But also, you know that when, for example, when I touch your skin, okay, you can feel it. Your skin contains like some cells, special, some receptors for responding to that stimuli. Or when you are eating, when you are smelling, okay, All the cells, most of the cells in the whole body, they contain their own receptors for responding to the stimuli. So it's not only the characteristics of native cells, okay? Characteristics of multicellular organism only? No, guys. We know that bacteria and viruses even, they are responding to the environment, okay. For example, when we get a disease, when we take the antibiotic, okay, bacteria can feel that antibiotic, or the bacteria can sense it. So it senses that there's a substance, there's an antibiotic, there's a drug in the blood or in the body, okay, so they will respond it. So it's also the characteristics of unicellular and multicellular organisms, okay. Regulation it means like homeostasis. It's not responding to the stimuli, or we can say regulation is maintaining the body at the optimum condition. So the answer for the question is A, okay. A is the answer for this question. All of the followings are true of the smooth endoplasmic reticulum, or we call it as or we call it SER, okay. Also we have RER, okay. SER it means smooth endoplasmic reticulum, and RER it means rough endoplasmic reticulum. We know that rough endoplasmic reticulum is covered by ribosomes. So one of the differences between SER and rough endoplasmic reticulum is the presence of ribosomes on rough endoplasmic reticulum, while we don't have them on smooth endoplasmic reticulum. So let's read all of them, but we have to pay attention. Question says all are true except one of them, okay. So there's one of the options is not correct. It connects the rough endoplasmic reticulum to the Golgi apparatus. Yes, sometimes it connects the rough endoplasmic reticulum to the Golgi apparatus. It detoxifies the cell. Yes, exactly. As an organ in our body that's responsible for detoxification, we have liver in our body that's responsible for detoxification. Also the cells, they have their special organelles, okay, which we call them SER, which they are responsible for detoxification. It synthesizes steroid. Yes, they are responsible for synthesizing these steroids. It manufactures protein. This is not true. RER, as I told you, because it's covered by the rough endoplasmic reticulum, it manufactures the proteins. While we don't have ribosomes on SER, so its function is not manufacturing the proteins. Instead, it synthesizes lipids or manufactures the lipids. So all they are true, okay, except this one, okay, D. So this is the answer for this question. Microtubules, guys. Microtubules, all we know that they are hollow tube that make up cilia and flagella, okay. Also they are found in the spindle fibers and centrioles. So the microtubules, which are the parts of the cytoskeleton, also they form hollow tubes that make up cilia and flagella. So I think it's clear, and all of you know that, and we studied. It's mentioned in your book that the cilia and flagella are made up of microtubules. 9 plus 2, guys. We have 9 plus 2. We have this term that we are using for which it's related to the configuration of microtubules in the cilia and flagella. Okay, but let me explain for you what we mean by the 9 plus 2. It means if we take a cross section from the cilia and flagella, okay, we can find 9 pairs. Okay, this first pair, second, third, fourth, fifth, sixth, seventh, eighth, and 9. Okay, we have 9 pairs of microtubules that are found in the peripheral region. Okay, with 2 single microtubules in the center. Okay, so this is the cross section of cilia and flagella in which they are arranged in this way. Okay, 9 pairs in the peripheral region. Peripheral means in the border region, we can say, and 2 microtubules, but they are not in pair. Okay, that they are found in the center. So the answer for the question is A, configuration of microtubule in cilia and flagella. According to the theory of Endo Symbiosis, okay, let us see what we mean by the Endo Symbiosis. Autotrophic cells were the first to evolve. Autotrophic, it means producers, the organism that they make their own food, but this is not related to the endosymbiosis. Heterotrophic, it means consumers, or we can say the organism that they cannot make their own food. We call them heterotrophic. Okay, it says heterotrophic cells were the first to evolve, and it's not related to endosymbiosis. Chloroplasts evolved from mitochondria, and it says mitochondria evolved from chloroplasts. They are completely not true, guys. These two, as you see, these two organelles, they are different organelles, and they do not have any relation with each other. They have some characteristics that they share, but they are completely different from each other. So the answer for the question is E. Okay, let us read it. It says chloroplasts and mitochondria evolved when free-living prokaryotes permanently took up residence inside larger prokaryotic cells. So, endosymbiosis, guys, what you have to know is a theory. Okay, it states that it tells us in the beginning of life on Earth. Okay, chloroplasts and mitochondria are two different small free-living prokaryotes. Okay, but when the time is passed, they entered into the large prokaryotic cells. Okay, they took residence inside that. Maybe it's true or maybe it isn't. Okay, but just know that the theory of endosymbiosis related to the living of both of these organelles that take a residence, or they take a permission for living inside the large prokaryotic cells. Here we have a slide that contains a figure, as we see. Okay, the question asks, the circle below indicates the position of letter X in the field of your compound light microscope. We know that what all of us we use compound light microscope. If you want to get the letter X, okay, into the center of the field, it means if you want to bring it here, okay, what we have to do. So all of us, and maybe all of you or most of you, you used compound light microscope. Okay, the simple type of microscope that we used, that we have in the lab. So if we want to take this X into the middle, what we have to do, we have to like to move the slide a little bit to the right, okay, by that way the x comes to the left, and then we will move it to the down, okay, and by that way when we are moving the slide to the down it will move to the up, so the x will come here or it comes to the center. So the answer for the question is A, moving to the right and down, okay. All of the followings are correct about mitochondria except, okay, except they are the site of cellular respiration. Yes, we studied that and you know that, and cellular respiration takes place in mitochondria, okay. They contain their own DNA, exactly, guys. Mitochondria and chloroplast, they are these organelles. Both of them, they have their own DNA and their own ribosome. Okay, they are the site of photosynthesis. All it's very clear that this is the answer for the question. Photosynthesis process, guys, it takes place in chloroplast, okay, it takes place in chloroplast, not in mitochondria. They can self-replicate. Yes, they can replicate themselves because they contain or they have their own DNA, so they can replicate themselves, okay, according to the need of the body, and chloroplast too. They contain an outer double membrane. Yes, they are double membrane organelles. Also, chloroplasts are double membrane organelle. So the answer for the question is C. Okay, they are the site of photosynthesis. Okay, all the others are true. So guys, in this question, it says all of the followings are correct about plant cell, except, okay? So, A, it says they usually contain one large vacuole. Yes, plants contain one large vacuole, while in animal cells, we have many small vacuoles, okay? They are more in number, but they are smaller. Lysosomes contain hydrolytic enzymes for intracellular digestion. Generally, plants they do not contain lysosomes and centrioles. Okay, these are among the main differences between plants and animal. So this is the answer for the question, but we have to read the others. It says they store pigments in chromoplast. Yes, we have chromoplast that found in the flower part mostly. Okay, and they store other colors, and we have chloroplast that it stores green color. Okay, because it contains chlorophyll, while the chromoplast is responsible for the other color like red, orange, those colors. They have secondary cell wall in addition to primary cell wall. Yes, plant cell they have secondary cell wall. They don't have centriole. They have no centrioles. As I told you, plant cells they don't have lysosomes and centrioles. So the answer for the question is B. Okay. Which of the following involves growing living cells? So, in the lab, we use tissue culture process for growing the living cells, and we have cell culture, or we have a culture generally that's used for growing bacteria, and we put them in an instrument which we call it incubator. Okay, freeze fracture and freeze etching technique. Those techniques are used for studying cell membrane. Cell fractioning that's done by centrifuge. centrifuge is used for separating substances according to their densities, okay, which it is used in the labs for separating serum and blood or plasma, and it's used for separating any type of the material that's found in a tube, in a solution. Tissue culture, as I told you, it's like putting the cells or microorganisms in a petri dish, in which that petri dish contains necessary main component that's important for the growing of the cells or tissues. Okay, and then we put that petri dish into the incubator. Apoptosis, guys, it's not related to all of this. It's completely different, but you have to know that apoptosis is programmed cell death. Okay, so the answer for the question is tissue culture. Okay, and that's the last question in this chapter. So if you have any questions, you can write in the comments below, or you can contact me via email or my phone number. You can write to my Viber. I hope you took benefit from what I teach you in this chapter. See you all in the third chapter, which is about cell division, mitosis, and meiosis.